This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1247, recorded on August 22nd, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. We have 92 degrees and sunny here. We have a typical Austin summer day, and I'm done with that. But I still got to wait a month or two.
Oh, here we're back down into the 20s. It really cooled off last week. I don't know what it is right now. My phone is charging. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 80 Fahrenheit, which is 27 Celsius, and we have blue sky with white puffy clouds. Pretty nice.
It's nice. We have a guest for you today. He is an emeritus professor at the Wistar Institute and the University of Pennsylvania, Stanley Plotkin. Welcome to TWiV.
Thank you.
Good to have you. None of us have met you in person, so we're all going to learn about you today. And folks listening, if you like these programs, as I always say, please give us your support. We are a nonprofit. We don't do advertising, so we depend on your contributions to make these science shows all about science, truth, no lies, no disinformation. Go to microbe.tv/contribute. Stanley, I wanted to start with your history. Go as far back as you can remember. You grew up in New York City, right?
Yes.
And I know you went to Bronx High School of Science.
Yes.
Was that the one on Lexington Avenue?
Well, I'm not sure where it is now, because when I went there, it was in an old building and has since moved.
Yeah.
But it was a critical experience in my life and undoubtedly of many other people's lives. I cannot overestimate how much it influenced me and many other people. And I hope it is still doing that. It's an example of the kind of high school that should be available for all young people who want to enter into learned professions.
So you must have had at least an inkling that you were interested in science in order to enroll there. Is that right?
Well, yes. I would say so. I entered, actually, I guess I was 12. Yeah, I was 12 years old. And by that time, I knew that I wanted to enter some sort of learned profession, so to speak. And New York City gave an examination to primary school students, and the results of that examination allowed people to enter specialized high schools, including the Bronx High School of Science, which was in the Bronx, where I grew up. And it was actually at age 15, when I was at Bronx High School of Science, that I read two books that forever influenced my life and made my decision. The two books, which I'm sure are familiar to you, are Arrowsmith by Sinclair Lewis and Microbe Hunters by Paul de Kruif. And once I read those books, I had no doubt about what I wanted to do in life.
Yeah, I think we've all read them both, right?
Yes.
I've read Aerosmith. I'm not sure I've ever read Microbe Hunters. I need to do that.
You should do it. It's good. It's very good.
Aerosmith was many, many years ago, but, good.
By the way, Bronx High School of Science is still in the Bronx. My error. I was thinking of Hunter College High School,
Mhm.
which is in Manhattan, and that's a high school that's run by Hunter College,
Yes.
and it's a very good school. No tuition,
Mhm.
and a very good school.
I feel like we've interviewed other people who went to Bronx High
Bronx.
School of Science. Were there are any other luminaries of our time that you went to school with, Stanley, that you can think of?
Luminaries. Oh,
Besides yourself, of course.
Besides yourself, yeah.
Well, I can't think of anyone else in my class that has achieved renown, but of course, I'm sure they went into many different professions, not just science. But it seems somehow unlikely, but it was such an intellectual atmosphere. That is, the students were competing, the teachers were first rate. They were all well educated and in a way certainly brilliant in the teaching profession. So I cannot overemphasize how important it was to me and to many other people who grew up in New York.
So after Bronx, where did you go to college?
Well, so New York State had a scholarship program that is based on an examination. They would financially support college. And I should add, subsequently, there was an examination to support college. And so I took the exam. I got a score high enough to get into Bronx Science and subsequently New York University. And I emphasize that because, and eventually medical school, but I emphasize that because I don't know that my parents could have afforded college and medical school. So I owe a lot to the state of New York.
So you went to New York University as an undergrad, right?
That's right, yes. At that time, the campus was in the Bronx, on the banks of the Harlem River, overlooking the river. It was a beautiful campus. I think now it's some other college is using it. But anyway, it was a great experience, and then medical school.
So you went to NYU Medical School also?
No, not NYU. It was the Downstate Medical Center in Brooklyn. Yeah.
Okay. And was it your intention to practice medicine?
Well, not to practice medicine. As I indicated, after reading those two books, I knew I wanted to do research, but and indeed I did think about taking a PhD rather than going into medicine. But my father, who was obviously more practical, said I should take the exam for medical school because I'd be better off with a medical degree. And so he convinced me to do that, somewhat against my feelings at the time. And I remember distinctly when the letter came saying that I had won the admission to medical school. I remember distinctly because my father opened the letter before I did, and came bursting into my room to tell me about this. And I remember saying to myself, "The die is cast," and so I went to medical school instead of taking a PhD.
But did you, aside from maybe an internship, did you ever actually practice medicine, or you quickly wound up in research?
Well, so after internship, which incidentally I took at Cleveland because John Robbins, one of the Enders, Wellers, Robbins group, was the head of pediatrics at Cleveland Metropolitan. So I applied there, thinking that I would learn something from him, which I did. So after the internship, I applied for a residency, where was I resident? Well, I came back to Children's Hospital of Philadelphia for my residency, and that was also a choice because Joseph Stokes was the head of Children's Hospital of Philadelphia. The name may not ring a bell to you now, but he was an early clinical researcher in vaccines in Philadelphia. So I was fortunate in that the things that I wanted to do, I was able to do.
So somewhere along the way, the Epidemiologic Intelligence Service fits in here. Where's that fit in?
Yeah, well, okay, so after the story there is that after or during my internship, at the time there was a draft, and I had signed up to join the Air Force because I wanted to learn how to fly, which I did later in life, but that's another story. But one of my fellow interns told me about the CDC and the Epidemic Intelligence Service. And I said to myself, well, that might be a way of getting into public health. So I got free from the Air Force. They allowed me to apply to CDC, which I did, and joined the Epidemic Intelligence Service, and went to Atlanta for training. But luck is everything in life, chance. And when I was after training in Atlanta, there were different assignments. One of them was to Philadelphia to join an anthrax investigations unit. Now, did I have any interest in anthrax? No, of course not. But by that time, I had been reading the virologic literature, and I read papers by Hilary Kaprowski, who had just become the head of the Wistar Institute in Philadelphia. So I said to myself, if I go to the Wistar Institute and join the anthrax investigations unit, I might be able to meet Kaprowski and get into his lab. And lo and behold, two things happened. One, I don't know if you want to get into this, but one, I did join the anthrax investigations unit, and weeks after I arrived, there was an outbreak of inhalation anthrax in Massachusetts. I can come back to that if you're interested. But at a certain point, about a month after I got there, I went into Hillary's office and asked him if I could work in his laboratory. He sort of looked me over and said, "Yes, okay." So that got me into his laboratory, and where I started to work on poliovirus. As you probably remember, his was the first oral polio vaccine. And, well, I can get into that if you're interested.
Yeah, I'd like to. So these were the attenuated polio viruses that were competing with Albert Sabin's, right?
Yes, exactly.
So what role did you play in that?
Well, so as you know, Hillary had developed the first attenuated polio vaccine. And he was starting to test it clinically. But he had started with a type 2 strain. And he needed type 1 and type 3 strains. And so I started to work in his laboratory on attenuating polioviruses, which could be used clinically. And eventually, he started clinical trials in Poland, where he was born, and in what was then the Belgian Congo. I can get into that more deeply, but anyway, so I learned how to attenuate polioviruses and grow viruses in the laboratory. And at a certain point, he asked me to go to the Congo to collect data on his vaccine, on the use of his vaccine, which I did. And that's, again, a long story where I learned a lot, not only about epidemiology, but about the situation in Africa, and was escaped being strung up by a mob, and all sorts of things. But anyway, so through him, or through his lab, I learned virology, epidemiology, and immunology, I guess you would say. So I don't know if you want me to go into any detail, but
so I'm. Pardon me. I have to ask. At this point, you must be in like your early 30s or something like that. Late 20s, early 30s.
Yeah.
Okay. Just trying to sort of put myself in your shoes. How exciting! Very cool.
So, Stanley, as you know very well, the Sabin vaccines were the ones picked, and right away we learned that they were genetically unstable. They could cause polio in recipients. Was that a property of the Kaprowski attenuated polioviruses as well?
Oh yes, definitely. I mean, it was clear that back mutation would occur with the early strains. I don't know if one can say that the Kaprowski strains were any less likely to mutate. Probably not. I mean, there are not enough data to say that there was a difference between the Kaprowski strains and the Sabin strains. My guess is probably not, that they would mutate as well as the Sabin strains in the gut of vaccinated individuals. But we never really tested that possibility.
In all cases where, I'm sorry, Kathy, go ahead.
I was going to ask, why the Sabin one is the one that we know about today. Was it ahead chronologically in terms of those clinical trials versus the ones in Poland and Africa, or did it have different outcomes?
Well, of course, to my knowledge, there's no head-to-head comparison, so that makes it difficult to answer. But my guess is, because of my familiarity with the Kaprowski type 1 strain, that there probably wasn't much of a difference. I would guess that given enough vaccinations, there would be back mutations of the Kaprowski strain as well as the Sabin strain. But the type 1 Kaprowski strain was damn good. I mean, it was immunizing and well tolerated, and I showed in the Congo that it was effective. So there never will be a head-to-head comparison, unfortunately. And also it has to be said that Kaprowski's type 3 strain was, which he got from forget his first name, Fox in New Orleans.
Fred Fox?
Was just a grab, and probably was not as good as the Sabin type 3.
I thought that Joe Melnick had compared them in monkey neurovirulence test, right?
Yes, he did, and I think his judgment was that the Sabin strains were more attenuated, but I don't think there was much of a difference, at least for the type 1.
Okay.
I think they were both quite good.
So, as you know, in the early 60s, we switched from IPV. So Jonas Salk's IPV had been licensed in 55, and then we switched to OPV. Do you think in retrospect that was a good idea? Because OPV has been associated with lots of cases of vaccine-associated paralytic polio.
Well, so here I'll be quite blunt, because strategies should change according to the facts. Mm-hmm. And the Sabin strains, oral polio vaccine strains, have achieved tremendous results in suppressing polio throughout the world. And there's certainly no criticism of that. But things have changed. We now have eradication in much of the world, with the exception of Africa, or Afghanistan, etc. So, in my current view, and I think many people agree with this, we should be switching to IPV as much as possible. Mm-hmm. And I've written this. I mean, have papers or participated in papers saying this, that we should be extending IPV as much as possible to prevent cases caused by oral polio vaccine. It's obvious. So I have not been happy about the WHO strategy. And I think they're backtracking about it also at this point. So I think one has to change strategies according to what the epidemiology tells you. And at this point, IPV will protect against vaccine-caused paralysis, and therefore it should be extended as much as possible.
So maybe we'll get back to polio in a bit, but getting back to your career, at some point you started working on rubella vaccines, right? Can you tell us all about that?
Yes. Well, so again, when I started out, measles vaccine had just started to be used and obviously was being very effective. And it was obvious that there were other diseases that needed to be controlled, one of which was rubella. Now, again, chance. After or during my residency in pediatrics at Children's Hospital of Philadelphia, they had an exchange program with the Great Ormond Street Children's Hospital in London. And the head of the virology laboratory at Great Ormond Street was a fellow by the name of Alastair Dudgeon, obviously British, but a very high-class British Brit. And he came to visit Kaprowski in Philadelphia, and so I met him. And I, by that time, knew that I wanted to work on rubella, and I discovered that he also was interested in rubella and had a virology laboratory. So, and again, just about that time, the first isolations of rubella virus took place in Boston and Washington. And so that there were ways of isolating rubella virus in cell culture. So I took a year at Great Ormond Street training in pediatrics, but at night I would go to Dudgeon's laboratory and work on cultivating rubella virus. So that was, let's see, that would have been 1962. In the spring of 1962, I was reaching the end of that residency in London, and clinically we began to see patients with rubella, and which was maybe not entirely surprising, but essentially an epidemic of rubella started in Europe. And when I came back to Philadelphia, it followed me, and cases began to occur throughout the United States, including Philadelphia. So by that time, I knew how to cultivate rubella in the laboratory, and Kaprowski gave me a laboratory at Wistar. And so I began to work on rubella virus with the idea of developing a vaccine. But the first year was the height of the outbreak in the US, which involved millions of people and thousands of pregnant women, including many in Philadelphia. And I was the only resource, became the only resource in Philadelphia for diagnosing rubella in pregnancy, and then subsequently, of course, in infants. And so I spent a year or two doing clinical virology as well as starting to work with the virus in the laboratory. And that was a very sobering experience. I mean, seeing the anguish of women who had to decide whether to abort or to carry the infant to birth. It was quite an experience.
For the listeners, this is the big danger of rubella is infection during pregnancy, right? And effects on the fetus or the newborn.
Yes, particularly during the first trimester. After that, the risk goes down quite a bit, practically to zero. But during the first trimester, the risk, depending on the time, etc., is upwards of 1/3. It depends on which study you're looking at, but it's a very high risk during the first 3 months of pregnancy.
And the result is a spectrum of pretty severe birth defects, correct?
Yeah, well, so it ranges. Infants, some infants, well, there are spontaneous abortions caused by the virus, but some infants are born with neurological, cardiac, other severe abnormalities, cardiac malformations, and of course attacks on the brain, and a variety of other abnormalities. So there are severe cases, and there are milder cases, but those who are affected are significantly affected. And of course, there's mortality as well in the infants. So it was a very sobering experience, I can tell you that.
So I presume you took your experience with attenuating polioviruses to attenuate rubella virus, correct?
Exactly. Exactly, which had been my intent, but chance, and the outbreak was a bad chance for everybody else, but it was quite fortunate for me.
And so then tell us a little about the WI38 cell strain, which I've had the pleasure of using in my lab in the past, and how that fed into making the vaccine.
So that, yeah, so Leonard Hayflick was also at Wistar. And so, we became co-workers, and when the issue came up, what cells should I try, in what cells should I try to attenuate rubella virus, it seemed to me that the safest cells would be one of Hayflick's human diploid cell strains. And this was because everyone had experience already with contaminating viruses in monkey kidney and other animal cell strains. And so I thought that the safest would be to use a human embryonic cell in which to attenuate rubella virus. Well, we can get into that if you want, but I mean, that became a, what shall I say, a very controversial choice, and got me into lots of arguments, which I can tell you about, including with Sabin. But anyway, that was my choice, and I think I was right. Well, I know I was right because I can tell you about that if you're interested.
So the use of WI38 is because it's a diploid cell
Yes.
line, right? It's not
Yes.
like a helicell with aneuploidy. But the problem would be it's derived from human embryos, and then
Right.
people who don't like abortions don't want to get the vaccine, right?
Well, that did become an issue later, although the Catholic Church decided that the advantage was worth the possible curse, and so they have recommended a rubella vaccine in diploid cells.
So Stanley, the WI38s were used for the attenuation as well as the production?
Exactly.
Oh, okay.
And the attenuation, is that serial passage?
Yeah, serial passage and yes, and selection of the best growing virus. We eventually, I'm trying to remember exactly why, but it was the 25th passage that at that point it seemed that the virus was growing very well, and that and then clinical studies showed that it was attenuated. So that's where we stopped.
Yeah, so I was going to ask about those clinical studies. How did you assess attenuation?
Okay, so here I was in Philadelphia at Wistar, and I obviously needed to test the strains in humans. Well, it was easy to, to find adults, seronegative adults who were willing to take the experimental vaccine. But in addition, I actually went to the Archbishop of Philadelphia and said, we're developing this vaccine and we need to test it in children. This vaccine could prevent abortions subsequently in women infected with rubella, and he agreed. And so we did studies in children showing that we could induce good immune responses to rubella virus. And then, of course, we had to interest pharmaceutical companies into taking up the vaccine and testing it in larger groups. And that's a story in itself, because we ended up at that point going to Britain, because I had met the head of the British organization, which is parallel to the biological standards outfit in the U.S., and he was enthusiastic about the rubella vaccine, and so the Wellcome Company in Britain was really the first commercial producer of the RA27-3 vaccine. If you want to, I can go into the story of how Merck took it over, but that happened later, after it became clear that the RA27-3 was better than any of the other rubella strains.
So what's the status now of rubella? Is it possible that rubella could be eliminated? I think you wrote a paper on that later, actually.
Yeah. Well, so the situation at the moment is that rubella is pretty much gone from the Western Hemisphere, at least pending reintroduction thanks to Kennedy. But it is generally agreed that rubella is absent from the Western Hemisphere. It is also gone from Europe, except possibly from Poland, because the data are not sufficient. And I think Romania is the other country where it's simply not clear. But the other European countries are free of rubella. Also, Russia at least claims to be free of rubella. So what we're dealing with at the moment is Asia. China is making a lot of progress. India has started a campaign. I mean, they still have rubella, but they're making a lot of progress. I think Thailand is pretty much free of rubella. So that leaves Africa. And, needless to say, who knows? I mean, we can't, we don't even have a good idea about the epidemiology except for South Africa. But progress is being made. Senegal, for example, is doing quite well. So we'll see.
Is your vaccine being used in all these circumstances?
Yes, there is a Chinese. Well, there are Japanese strains of rubella. The Chinese, frankly, I think they're using RA27.3, but it's really hard to know. They just don't give a lot of detail.
Okay.
But the point being that, RA27.3 is produced commercially by a Serum Institute of India and another Indian company, and so there's plenty of rubella vaccine around. The issue is simply combining it with measles and getting it into children.
So currently it's part of measles, mumps, rubella vaccine, right?
Yes.
MMR.
Well, mumps is not widely spread in the rest of the world. That is mumps vaccine, but MLR is widely used.
I was wondering what you thought about all the attacks on MMR originating from Andrew Wakefield and continuing to this day, especially in light of the measles outbreaks in the U.S.
Well, needless to say, I think it's appalling. But, frankly, I think this is the growth of know-nothingness in the U.S., which is frightening. But I see it as part of an overall reaction, saying that that expert opinion is not important. What's important is my opinion, and so people are willing to accept nonsense, and in America, you can't stop that. It's a free country, so to speak. My European colleagues tell me that is not yet the case in Europe. So I have said this, actually, given a talk at a vaccines meeting, that I think vaccine research will move to China and India, and that's where the progress will be. And maybe Europe, but Europe doesn't have a big enough scientific apparatus.
So it seems like you've worked on a lot of viruses with respect to vaccines, and we could talk about rabies, but also I'd like to hear a little about CMV, which I know you've said has been your dream to see a vaccine for that. And I've kind of lost track of what the CMV platforms are and where the clinical trials stand, and maybe you could bring us up to speed on that.
Sort of the love of my life. So, as I just recounted, in the 60s, I was working on rubella, and by the 1970s, that was pretty much solved. And so, of course, I started looking for another target. And at that time, the importance of cytomegalovirus was just being discovered. And so in about, I think it was 1973, I started to work on attenuation of cytomegalovirus. Unbeknownst to me, two workers in Great Britain, Ellick and Stern, also had that thought, and so they tried to attenuate CMV. Their strain eventually was tested, and Merck actually studied it. To make a long story short, it was dropped. It was not sufficient. But I continued to work on attenuating CMV, and developed an attenuated strain and started clinical work. And from my point of view, it was getting good immune antibody and cellular immune responses. Well, so this comes a long story. One of the things that I did was start testing in organ transplant patients. And as you know, particularly those who are seronegative for CMV and receive an organ from a seropositive donor, are at risk of severe disease and rejection of the transplant. So I started studies in them, and this is all published work. We had pretty good efficacy against transplant disease. But then, of course, the idea was, well, could we protect women during pregnancy? And so we started along those lines as well. But then, so this was the 1980s, 1990s, and the problem was that people were concerned about using a live virus that could become chronic in women who might later become pregnant. I hope that's clear. And so the companies that I tried to interest, etc., became afraid of using a live vaccine. And so research switched to other approaches to a CMV vaccine, which, as you probably know, is going on today. Moderna is finishing a phase 3 trial with an mRNA CMV vaccine, and a couple of other companies are trying to develop candidate vaccines. And so the field is still active, but it's taken a long time, and that's because the biology of CMV is much different than the biology of rubella. As you know, it's a virus that's capable of becoming chronic, that the immune responses that control CMV are not simple neutralizing antibodies. The importance of cellular immunity is critical, and there are multiple antibody functions that arise after natural infection. So it's a much more complex biologic problem. I don't think that it's unsolvable. Au contraire, I think that there can be a CMV vaccine. The difference is going to be that because of the variability of challenge dose, we may never be able to have 100% protective vaccine. because women, for example, who have a child in daycare are challenged every day by the virus excreted by their infant. So, as we know from studies in primates, if you have enough challenges, eventually you will overcome immunity. So, I think we will probably never have 100% protective CMV vaccine, but I'm damn sure that we can protect the majority of women from serious CMV infection in their infants. I hope the trial being conducted by Moderna will show good efficacy, although we can get into the defects of mRNA vaccines.
Actually, I'm curious. This is a detail, but I'm curious. Is the Moderna vaccine, has it got more than one antigen in it?
No, basically, well, it has two antigens in it, and that does give good neutralizing antibody responses. But the question is, will the cellular responses be good enough? And as I just described, will it protect against large challenge doses? And by early next year, we should have an answer to that.
Now I'm curious what you think are the defects of the mRNA vaccines.
Well, I think it's pretty clear that persistence of immunity is one issue. And the second issue is the broadness of the breadth, I should say, the breadth of immune responses. Will you get enough cellular immune response to evoke memory and T cell responses that control replication if infection does occur?
Kathy mentioned rabies. I wonder, how did you get into developing a rabies vaccine?
Well, rabies was one of Hillary's primary interests. And so when I came to Wistar, one of the things he wanted me to do was to help show the efficacy of rabies vaccination. Now, I have no claim to the work in the laboratory that eventuated in the rabies vaccine that came out of Wistar, but I did work with Tad Victor in Hillary's rabies lab, and when they got to the point where animal studies had shown good immune responses to rabies and protection against challenge with wild rabies virus, he asked me to conduct, I guess, what you would call a phase 3 trial. Well, no, not really, because we weren't challenging people with rabies virus. But anyway, to show that there was good immune responses in humans. So I got involved in that, and it seemed that the obvious group that would be interested would be veterinarians in training. So I set up a study in veterinary schools throughout the U.S. And after getting an agreement from the schools, I started on a trip around the U.S., going from one veterinary school to another, vaccinating, giving rabies vaccine, and then, of course, arranging for blood collection, etc. And I can tell you that was an experience that I would not like to repeat, because veterinary schools are usually located in places that you would never visit. And so I remember one place where I couldn't get dinner because there was no place to get it. But nevertheless, the study did show good immunogenicity in the students, and led to the eventual licensure of rabies vaccine, which is tremendously protective and effective. And as you probably remember, in the old days, people were getting 21 or even 28 doses of rabies vaccine. And now they get 3, maybe 4, and probably don't need more than 2 or 3 to be protected, maybe indefinitely, but certainly for many years. So the whole thing has changed dramatically.
So this is an inactivated virus, is that correct? Instead of
Yes.
a attenuated virus? Okay.
Yeah.
So
there are
you've been
attenuated rabies vaccines, but obviously difficult to show safety in humans.
So you've been talking about prevention of rabies in veterinarians, but you can also use it after exposure, right?
Oh, yes, sure. And today, it's funny you should ask that, because this morning there was an email from a doctor in India asking me a question about the schedule, etc. So even in India, which of course has a major rabies problem, human diploid cell vaccines are being used and saving lives, I'm sure, left and right.
So why is it possible to stop rabies after exposure by vaccinating? It doesn't work with other vaccines, right?
Well, because the virus has to travel from the site of injection, probably through the nervous system. I mean, through nerve channels rather than through the blood, to get to the brain. And so everything depends on getting antibodies and possibly cellular immunity, although that hasn't really been studied very well. But anyway, inducing antibodies, and maybe this indicates that blood transmission is important as well, protects the brain against the introduction of rabies virus. And it seems that the virus has to replicate locally before going to the brain. I say that because, as you imply, starting a vaccination days after the bite still seems to be effective, although it gets less effective the longer you wait. But I think it's pretty clear that there has to be local replication before the virus travels to the brain. And starting immunization, within, let's say, certainly within a week of the bite, is highly protective.
I'm curious, once the virus enters the nerve and gets into the brain, does it further replicate in the brain? Do you know?
Oh, yes.
Yeah.
Yes, definitely, yes. Yeah.
Okay, and that's results in all the symptoms of rabies.
Sure.
Okay.
I want to switch to another virus, because you recently had a commentary in a column that it brought a smile to my face. It's called Stan Plotkin's Corner in Pediatric Infectious Disease Journal. And it's about West Nile virus, an American disease. And just like you had something happen this morning, they announced on the radio that here in Michigan, there's the first West Nile virus death this year from West Nile virus. And so just a reminder, this is the virus that became a little more on people's radar when Tony Fauci got it last August or so. But what's the status there on vaccines, and what are the hurdles for that? And who would need to get it in the United States? I mean, would you have a population like veterinarians for rabies to start with? Or
well, the first thing is that it should be renamed because, it obviously occurs outside the United States, but we should rename it the USA virus or the federal virus or something like that, because it is now indigenous to the United States and widespread. And, you know, I tell my wife, who does a lot of gardening, that she has to be very careful about not being exposed to mosquitoes and, you know, as well as ticks, because we have Lyme disease around here. But anyway, it is now an American virus. And there should be a lot more publicity about it, and a lot more anti-mosquito work, because it is a virulent disease, and we should be doing more. Did I answer your question?
Well, I'm wondering, you know, what kinds of things would be possible for vaccines or people
I think developing. I wouldn't say that I am very familiar with the experimental literature, but I don't think there's any reason why a vaccine couldn't be developed for West Nile. It shouldn't be a big problem. It's a question of market.
That's a good. So who would get it, right? That's what Kathy also asked.
Yeah.
Who would?
I think it would be appealing to people who do a lot of outside work.
Yeah.
You know, there's a lot of not just recreational people, but there are a lot of people who work outside.
Mm-hmm.
I should think that they would want the vaccine, but it's also, you know, here we get into the CDC issue, but one of the things that CDC is tasked to do, and which generally is done well, but needs to do better, is to inform the public. Now, here we get into politics, etc. But as someone who spent several years at CDC or working for CDC, you know, you have dedicated people who are trying to do their work, etc. And as you know, who published the MMWR.
Mm-hmm.
But I don't know that what they say gets sufficient spread in the press, and you know, I don't know that it has a sufficient impact, which it should have. But I don't know how to deal with this exactly. But somehow CDC's findings should be more prominent in the public eye than they are now.
Well, it seems to me that the goal of the current administration is to dismantle the CDC. So I'm not sure it's going to be ever an issue at some point, Stanley.
Well, I don't need to say that, especially at my age, that I am horrified to see what is happening to public health in this country. And the situation gets worse day by day. I was just reading that. I must say that I'm afraid now to listen to the news because it gets worse day by day. And what's happening at CDC is horrifying to me. The people who are being fired, the destruction of the public health apparatus, taking over the ACIP, which was a group of informed people, and populating it with people who don't know what they're talking about or are purposely against public health. It's horrifying. I feel personally insulted that this is happening in my old age, but not much I can do about it.
Seems to me that you have spent your whole career developing vaccines to protect the public's health, and now you're witnessing this incredible anti-vaccine sentiment, along with destruction of the public health apparatus, as you say. And I wonder, so this anti-vaccine position, why does it exist, Stanley?
Well, I have two comments on that. One is that scientific education in this country is insufficient. I've recounted my personal history, but I don't think many students are getting the basic information that is needed about health and science. And so you have a growing population of people who are ignorant about things that are basic to health and public health and to science. So one of the things that I advocate is that science education should start in grade school. That every child from the first grade on should be taught about what science is and how you evaluate evidence. And I don't think, I'm as an ex-pediatrician, I know how bright children are. And before they become prejudiced, they are open to learning things. And so that's where I would start, because now you have a lot of people who are making decisions who have learned prejudices without learning the basic facts. And I'm sure that's true of things besides science, but it is certainly hurting science. Now, the other thing is that there were no podcasts when I was a kid. And so now, I'll be blunt about this. Now, any jerk who has access to being conducting a podcast can spout nonsense, and there are plenty of people who will believe him because they're not listening to anything else. So this is the decline and fall of the American empire, because we're being besieged by nonsense. And I certainly don't know how to solve that problem, but it shows you how progress of civilization is not a straight line. It never has been. Sometimes it moves forward, and sometimes it moves backwards. And unfortunately, that's where we are today.
Well, Stanley, that's why we do this podcast. We're trying to counter those other nonsensical podcasts, but they get a lot of attention. You know, the truth doesn't sell very well.
That is true. Yes.
I'm interested in talking about science education because I think this is important. You talk about learning the basics, but you also mentioned learning basically how to evaluate things. I think critical thinking is the at the center of this, and I think all too often science education is just a blizzard of facts that can turn kids off. And I think they need to learn the process of science, the scientific method.
Exactly, I agree.
And it applies. You know, the scientific method is not just for science; it's for evaluating any sort of information that comes your way and trying to figure out where's the truth.
Mm-hmm. Yes. Yes.
So, Stanley, we skipped over something because you went to Paris.
We're not done yet.
I know, but we're chronologically, we skipped over Sanofi Pasteur.
Oh, yes.
Could you just briefly tell me where that came in your career and what you did and what the influence was on everything?
Right. So this brings us to 1990. So by that time, I had been running infectious diseases at Children's Hospital and of course, running a laboratory, actually two laboratories, partly at CHOP and partly at Wistar, where I was still a member of the faculty. And then something happened, which shows you how you could be stupid. Also, the chairmanship at Children's Hospital became vacant, and I applied. And make the long story short, I was not chosen, which made me rather peeved. In retrospect, of course, it was very stupid. But anyway, I was also offered at that point. I was offered a chairmanship in Los Angeles. I went out there with my wife and looked. They showed me a house on the Pacific, which has probably been destroyed in the last storm. But anyway, offered me this house, and I was seriously considering it. I came back to Philadelphia, and at about that time, Charles Mérieux, who was the head of what was then called Pasteur Mérieux, the French vaccine company, who knew Kaprowski, came to Philadelphia and offered me the job as head of vaccine development at Pasteur Mérieux in Paris. So, actually, I had taken a sabbatical at the Pasteur Institute before. I've been there, was there for about 7 months. So I knew Paris fairly well, but nevertheless, he flew me to Paris and showed me the place where I would work, which was actually Louis Pasteur's summer laboratory outside of Paris, and dropped me off at the Pont des Arts, the bridge across the Seine, in which you can see the Eiffel Tower on one side, Notre Dame on the other side, the the French Academy in one direction, and the center of Paris in the other direction. And I decided that I'd take the job in Paris. So so I came in and I spent 7 years as in Paris working for Pasteur Mérieux. And actually, it's funny, the French have a very strict age limit. At age 65, you have to retire. I didn't quite realize that, but I reached 65 in Paris and had to give up my job and return. But anyway, during the 7 years, it was quite interesting because I learned how industry works. And I learned both positives and negatives. Industry, on the positive side, can get things done because they have the resources, they have the people, and they can move fast if they want. The negative side is that they have to make a profit. So the economic issues weigh heavily in deciding what research is done. And the other thing which I discovered is the competition. Now, in academia, you have friends, you have colleagues. There may be some competition, but by and large, you're independent. You have colleagues. Sometimes you collaborate with them, but nobody is trying to undercut you. But in industry, it's quite different. There are people who want your job. And so you really have to be wary about that. Nevertheless, I enjoyed my time there and eventually, as I said, had to retire and come back. I must say, I regret now not having stayed in Paris, but my wife wanted to come back to the U.S. for family reasons, etc. And so we came back and gave up our residence in Paris.
One other thing that you put out there as a teaser when you didn't join the Air Force, that you had always wanted to learn to fly.
Yeah.
So you did go on to do that. Tell us about that.
Well, so actually it was after I came back from Paris. There's an airport that is, actually it's 4.5 nautical miles from here. And I went there and enlisted in a training program, and eventually got my license, and eventually bought a single-engine plane, actually made in Austria, but it's widely. There are many Austrian-type small planes in the U.S., as well as American-made, of course. But anyway, so I owned the plane and flew around a lot, enjoyed it. But then, I guess, 5 years ago or something, I decided that one thing that's essential is reflexes. You have to be ready to respond to an emergency immediately, almost without thinking. And I decided, I'm not sure I can do that anymore. So I sold the airplane, much to the relief of my wife. And I regret that in a way, but it was a wonderful experience. The leaving, I mean, this has been expressed by many others more poetically, but the feeling of leaving the earth, lifting off, is just incomparable. And you don't get that commercially. It's not at all the same. But flying yourself is really, really an experience.
To be a bird, yes. So, Stanley, I have a name for West Nile virus. You want to rename it, right?
Yes.
Maha virus.
Maha?
Maha, make America healthy again.
Oh, yeah, good. That's a good idea. Yes, that could fly, so to speak. Yes. Yeah.
So, Stanley, I read a commentary of yours where you looked at the costs of not immunizing neonates with hepatitis B virus vaccine. And, you know, I'm sure you know, RFK Jr. recently has said that we don't need to immunize neonates with that vaccine. So why should we do it?
Well, that's another one of his stupid ideas. The point is that you want the highest coverage possible. The longer having a child at birth gives you an opportunity. That is, every child gets born. And so you have the opportunity to provide a lifetime protection against hepatitis B. If you wait, then the coverage diminishes. And, you know, we don't have 100% coverage of measles, let's say, in this country, or other pathogens. And with a vaccine that works given at birth, you have the opportunity to provide virtually 100% protection for life. I mean, what could be better than that? And yet, Kennedy is trying to stop that. It's just another stupid idea.
Did you see recently a Danish study was released showing that aluminum adjuvants are safe? We actually covered it here on TWiV. And he's now, yesterday, he asked the journal to retract the paper.
Yeah, well, of course, I know the author of the paper, and as he pointed out, it was impossible to have an unvaccinated population in Denmark because everybody accepts vaccines in Denmark. But by comparing populations that had high vaccine coverage and low vaccine coverage, he drew a conclusion. Now, you know, I'm not against vaccine safety studies. I mean, one has to admit that nothing, whether it's medicine or vaccines, is without reaction. So the question is, what is the proportion of reactions compared to the advantage of being protected? And so what you do is you collect data. Now, as you well know, there have been many studies of aluminum-containing vaccines before. None of them has demonstrated to scientific accuracy that there are significant long-term reactions. But, you know, I'm not against any study that is well done So I would say if Kennedy is willing to finance a study that is scientifically valid, I would not be against it. It's relying on junk science that is objectionable. So, again, if somebody, I don't know how they would do this study, but if somebody has a population somewhere where there's an adequate control group to do a study of aluminum-containing vaccines, I wouldn't be against it. But to set up a study that is inherently biased is not only a waste of money, but it enables conclusions. It enables false conclusions to be made. So, again, one can't be against a good scientific study, but that's the sine qua non. It has to be a study that's valid.
It occurs to me that over time, if things keep going the way they are, the U.S. could become the control group for a lot of these studies.
Well, yeah, that's an interesting point. I suppose you could compare the U.S. with other countries, but that would have problems also. I think what can be said without hesitation is that there is no widespread or obvious significant problem with aluminum-containing vaccines. Now, is there, some 1 in 10,000 reaction that occurs? I cannot say. All that I can say is there's no evidence for it. Mm-hmm. So, if he were willing to set up a study of millions of vaccinations with an adequate control group, I don't know how he could do that, but if he could do that, I would support it. But just condemning aluminum-containing vaccines without any evidence is obviously going to contribute to the problem.
So, Stanley, he says all the evidence is there, but it's covered up by the FDA and the CDC.
Well, that's bull. I mean, that's nonsense.
So, another thing I wanted to ask you about, what's related to what we've been saying. So we're seeing declining vaccination rates in the U.S. We've just gone through a measles outbreak, still ongoing, the biggest since it was declared eliminated. Do you see other infectious diseases like polio coming back in the coming years?
Well, I think it's possible. In the case of polio, it would require somebody from Africa, basically, or Afghanistan or Pakistan, perhaps, coming into the U.S. and infecting other people. It's possible that might happen. But I would say the risk is low. It's not zero because that is happening in Africa. That is, people are carrying polio from one country to another. Whether it comes to the United States is a matter of chance. But that's because polio is now relatively rare. But something like measles, or rubella for that matter, could certainly come back to the U.S. because there's enough of that elsewhere in the world, so the chances are that somebody infected will come to the United States. And, particularly in places like Texas, if that individual comes to a group of poorly vaccinated people, then the agent will be reintroduced into the U.S. So, as the population coverage drops in this country, it is inevitable that diseases that have disappeared will come back. It's just a question of how prevalent those diseases are elsewhere in the world. The more prevalent they are, the more likely that they'll get into the United States. Polio is fortunately relatively rare.
So Paul Offit, who you know, once asked Maurice Hilleman, who you also know, what would it take to get rid of anti-vaccine sentiment in the U.S.? And he said, unfortunately, many deaths will have to happen before that occurs.
Yeah, I think that's true. By the way, you know, Paul was a student of mine.
Yeah.
And I think that's probably right. Unfortunately, I think that it will take reintroduction of disease before people wake up. And fortunately or unfortunately, that takes time. I mean, for the immunity level to decrease sufficiently. So it may not happen this year, but it will happen. There's no doubt about that. And maybe that's the only thing that will change public opinion.
You've written a little bit about vaccine mandates, which we saw during COVID, for example. I think there was a measles outbreak in Philadelphia years ago where vaccination was mandated. What are your thoughts? Are mandates useful, or do they lead to anti-vaccine sentiment?
Well, that's, of course, a very good question. And it gets us into the field of ethics. You know, this sounds like the old guy I am. But when I was a kid, there was definitely the feeling that you were responsible for your neighbor, that you had some public requirements. That is to say, that you had to think about other people as well as yourself. And that seems to have disappeared. So the argument that you are not only protecting yourself, but protecting your neighbor doesn't seem to have any value at this point. But, you know, when Sabin vaccine became available and my parents and their friends were all anxious to have their children vaccinated, there wasn't an issue, but now there is, because in effect people say, well, I don't need to be vaccinated. I can, you know, let my neighbors do that, and I'll be protected. So I don't know what to do about that. Teaching ethics in grade school may not be easy, but should be done.
You've also written that not having a Lyme vaccine is a public health failure. So why don't we have one?
Well, that's an interesting story. As you may remember, there was a licensed Lyme vaccine in the 1990s. And actually, at that point, I was at Pasteur Medio, and I say that because there was a Pasteur Medio vaccine and the GSK vaccine. And for reasons which were really trivial, they made a deal, and the GSK vaccine got licensed. And it worked. It worked pretty well, actually. But then, particularly in California, I think it was, there started to be agitation about the vaccine, that it was somehow dangerous and that people shouldn't take it. There were, and there were, to be honest, there were some joint reactions to the original vaccine. They were relatively mild and transient, but nevertheless, there were some joint reactions. I would say the evidence that they were related to the vaccine was not very good, but there was significant evidence. So anyway, a lot of agitation developed against the original GSK vaccine, and eventually, because of lack of purchase of the vaccine, GSK took it off the market. And so what is happening today is, in a sense, a replay of what happened years ago, and I hope with a different outcome. That is, more use of the Lyme vaccine that is almost through phase 3.
Is it?
Anyway, I will certainly give it to my wife, who does gardening, as soon as it's available, because I've seen cases of Lyme disease. It's not pretty.
Does anybody else have anything?
I'm good.
I'm good.
I'm going to ask 2 more questions. You talked earlier about new kinds of vaccine technologies. In fact, you've written articles about it. And I don't know if you saw this, but a few weeks ago, an article was published showing that you can deliver vaccines on dental floss.
Mm.
Did you see that, Stanley?
I didn't read the article, but I saw an allusion to it. but I don't have to tell you that the advances in vaccine science from when I started, well, I guess this is true of any scientific field, are enormous. You know, when I started, there were essentially 2 ways of making a vaccine. Either you attenuated the agent or you killed it. That was it. Mm-hmm. Now we have many different ways of making vaccines, and as you know, getting into cancer prevention and so forth. Well, of course, that's not surprising for scientific advancements. But my point is that the technology of developing vaccines is now fantastic. I mean, we can make vaccines in many different ways if we wish, but the utilization of vaccines is the issue, not the development. I don't think there's, well, maybe HIV is still a big headache, but I don't think there's anything that we cannot control, if not eliminate, by vaccination in principle. But it takes work and money and the enthusiasm. And that is what I'm afraid of, that is going to be decreased, underfunded, and therefore that vaccine development will decrease.
So the study that Vincent brought up is one that I mentioned on a previous episode, and it was mostly mouse studies. And so the remarkable thing was showing how it took 2 guys to actually do the flossing of the mouse. But I think the gist of it was, for my take home from it was that, there are a lot of reasons why people don't want to take vaccines, and shots are a big part of that. And if there's some other method of delivery, like the microdermal skin patches, or maybe something you get at the dentist's office if you're not afraid of going to the dentist, might be. So I think you're right. There's lots of ways to make vaccines and develop them, but still you have to get it over the goal line, as it were, and into the person. And so that's where that came up.
No, you're right. And the issue of DNA vaccines is an example of where delivery of the vaccine may be a major impediment to the development. I've been associated with a group at Wistar that's worked on DNA vaccines, and they've had a hard time because of the delivery issue.
Is that because you need a gun to deliver it?
Yeah.
And it hurts, I guess, right?
Well, I don't think it hurts that much, but it's been an inhibitor to
I can imagine.
use. Yeah.
That big coming at you with that gun is kind of scary. So you mentioned HIV. Are we going to have an HIV vaccine one day, you think?
Well, of course, making predictions is always dangerous. I think that we could have vaccines that ameliorate the course of disease. Preventing infection in the first place is a big ask, because, and that's something that I should have said earlier, that the biggest lacunae we have in vaccine development is mucosal immunity. How to induce mucosal immunity. We simply do not have enough knowledge. And that, of course, applies to sexually transmitted diseases, as well as others, but in particular, and to a infection or a challenge which may be repeated. That is, not just, an occasional exposure, but virtually everyday exposure. And we do not have ways of inducing mucosal protection. So I think we can have an HIV vaccine that reduces the replication and extends the lifetime of people, etc. But developing a vaccine that protects the mucosa against repeated challenge, that is a major problem. And we need to know much more about mucosal immunity in order to solve that. And I think that, that applies to other infections as well, but particularly to a sexually transmitted infection.
Stanley, if you still had a lab today, what would you work on?
Oh, boy. Oh, gosh. Well, I would say honestly that I would have to go back to school because, knowledge has multiplied and keeping up is really difficult, especially when you get old and you're not as smart as you used to be. But, well, I would like to see my work on cytomegalovirus end with a vaccine, and I think that is feasible. So I would like to see the end of that, so to speak. And I do think that HIV is still a number one target. And how to induce mucosal responses is an issue. One thing that I have been charmed by, if that's the right word, is the work of the Iwasaki lab using parenteral injection followed by mucosal exposure as a way of inducing mucosal responses. I think that's something I would like to see pursued more for mucosal diseases, which could include HIV as well. Prime the immune system and then come at it with a mucosal antigen and try to get the mucosa to respond.
With polio, if you give oral vaccine and then you go in with IPV, you get a mucosal boost.
Yes, that's right. Yes, yeah.
So maybe similar mechanisms, except different timing there.
Yeah, I agree.
Well, someone asked me the other day I don't have a lab anymore either, what I would do if I had a lab, and there's too much to think. There's so much. And we cover so much here on TWiV that I love. I just said polio because it was the easiest answer. Because there are a lot of unanswered questions with polio as well. But we can't work on it anymore. Okay, that's TWiV 1247. You can find the show notes at microbe.tv/TWiV. Questions, comments, picks of the week, TWiV at microbe.tv. And if you like these programs, please support us, microbe.tv/contribute. Our guest today from the Wistar Institute and the University of Pennsylvania, Stanley Plotkin. Thanks so much for your time, Stanley.
You're welcome. Good luck.
Kathy Spindler, Professor Emerita, University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun, and I really enjoyed getting to talk to you, Stan. It was just remarkable. Thank you.
Thank you.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time, and me also, Stanley. It's great to have this time to talk to you. It's a privilege.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.